My name is Sidi. I'm an assistant professor at Mount Sinai here in New York City. What I try to tell students about the job of a scientist is that it is 99% frustration, but that 1% of the thrill of discovery makes it worth it. This project addresses an important and often overlooked problem in cancer treatment, which is the resistance of patients to radiation therapy. 50% of the patients do not respond to radiation therapy, and if the tumor comes back as a result, unfortunately at this time, it is the equivalent of a death sentence. Looking at many cancers such as head and neck cancer, where radiation is a frontline standard of care, 50% of patients resist radiation treatment. And very often, if you look at the genomes of these tumors, these genomes have a mutation in p53. We use zebrafish in our research as a platform for novel anti-cancer drugs. We are fortunate that we have developed in zebrafish a p53 mutant line of animals that lack this tumor suppressor protein. And so here we have a very simple, straightforward model of radiation resistance in cancer. We can now use this model as a platform to throw drugs or mutations on this model and try to identify compounds, for example, that would restore a normal response to radiation in these mutant zebrafish. If we can find such a compound, we hypothesize that this compound could potentially be used in patients that also have a p53 mutation in their tumor and that resist radiation. If these patients now take this compound will resensitize the tumor to the radiation therapy. Our hope is to first understand how this new candidate therapy that we've identified works in cancer cells to make them change their mind. Instead of surviving radiation, we are going to die in response to radiation. So that's one aspect. The second aspect is to have an impact not solely on a fraction of cancer patients within a tumor type, but have an impact that transcends tumor type, broadens the spectrums of tumors that we look at, and help a greater population of patients. It's through support such as that provided by the Pershing Sohn Prize that we can now actually do the experiments and figure out for ourselves if the compounds we find in our fish model will eventually work in humans.